Hi, I'm Lisa Tiemann. I'm a soil biologist at Michigan State University. When I was in high school, I had two things that I really enjoyed. One was music. I was a trumpet player. The other was science, and in particular biology. When I was looking at schools and thinking about college, I looked at the University of Wyoming and was actually intending to be in their forestry program. But before I ended up at University of Wyoming, I decided that actually what I wanted to do was pursue my other love, which was music. So I ended up at the University of Wyoming as a music performance major. What I really wanted to do was be an orchestral musician and win a job as a trumpet player in an orchestra. But that field is so competitive. It's hard to get a job at a research institution as a professor, but it's probably 10 to 100 times harder to get a job as a musician in an orchestra. So I was first excited about soils when I found out about the nitrogen cycle. And I found out about the field of soil biogeochemistry. So nitrogen cycling is an extremely complex and really interesting process. And from that point on, I was kind of hooked with soils. And really what soil organic matter is sort of the cornerstone for plant productivity, and particularly in agricultural systems. As we harvest crops from soils and all of that material is removed from the system, we can deplete soil organic matter reserves. My background in music helps me with my creativity, and I think there's also some structural elements to music that help keep me organized and focused and thinking about data in different ways. So technology actually has a really huge impact on my career in the way that I am doing science. And the primary piece of technology that has really impacted the way that my entire field does science is really the molecular technology that we have now. The ability to take DNA and amplify certain regions of it so that then you can take that amplified gene and sequence it or do other things with it. Or now we can actually take all of the DNA from a soil and sequence it so that we can know all of the genes and all of the organisms that are present in a single soil sample. Now, 10, 20 years ago, we couldn't do that at all, and we had no idea about the diversity and the complexity of a soil community. Now we are just starting to take some of that sequence data and some of that understanding that we can get from these new technological advances and starting to couple that with some of the functional assays that we do so that we can really start to understand what it means if we have certain members of a microbial community in a soil. What does that mean? What are they doing? And what does that mean for functions on a larger scale? So if I have a certain microbial community in an agricultural soil and I see that nitrogen cycling is doing something particular, is there a connection between that particular microbial community and what we see happening with nitrogen cycling? And so being able to couple those two things to give us a better understanding of how certain processes that take place in soils are occurring is really going to help us to manage soils better and to manage things more efficiently in the future. But I would say if you have an interest in science, just explore and find out all of the different possibilities that there are, because there's so many.